Okay, so here we go. I'm back to school. I've been having so much fun with these video clips and stuff. I'm hoping they're working. If I need to pause, by the way, it's because I gotta go unarm the school again because it's 12:31. Anyway, so hopefully I'll get it done before then. You're gonna need for this, we're gonna do this little review thing, as you can see up over here. You're gonna need red. If you want to do the same color as I am, you're gonna need like a blue and then pink. So we're gonna go through this. We're gonna see how it goes. Hopefully it'll go good. And I think that's it. So here we go. All right. So if we look at this, we have, and we're going to start on the side that's aerobic cellular respiration. So these are those review pages. So one of the first things is, of course, we're talking about aerobic cellular respiration, which of course means that it requires oxygen because it's aerobic, right? But not all of the parts actually need oxygen. There's also the other option for cellular respiration, and that's anaerobic cellular respiration. And that's where you're going to do the same basic first part, right? It's going to be glycolysis, respiration. Okay? But after you do glycolysis, you're going to do one of two things. You're either going to do it as far as like if you're an organism that does this anaerobic cellular respiration, you're either going to be like maybe vertebrate muscle cells. So you're going to do lactic acid fermentation. Now, lactic acid fermentation can also be done by some bacteria. It's only going to get them, right, 2 ATPs, and it's also going to get you 2 lactate, okay, also known as lactic acid. The cool part is if it's our vertebrate muscle, we're in oxygen debt at this point. Not so cool. But the nice part is if you put lactate in the presence of oxygen, it'll then convert back to pyruvic acid that can then be converted into acetyl-CoA right here through changing to acetic acid. Okay? The other option is, say, your yeast. You can do alcoholic fermentation. Okay, alcoholic fermentation can be done by some bacteria and definitely by fungi. Unicellular yeast is an example. You're also going to get 2 ATPs. You're going to get, well, helps if I can write that correctly. See, it still even works on Bento. ATP. You're going to get 2 ethanol, also known as ethyl alcohol, hence alcoholic fermentation. Ethanol, like the stuff that they add to your gas tank now. Okay, but you're also going to get 2 CO2s, okay? So it's still going to produce CO2 just like we do because they need to get rid of that excess carbon. And that's also why it raises the bread. And you let the yeast digest all those starches that are getting broken down into glucose that it can then go through and convert to pyruvic acid, then go through alcoholic fermentation to make its own food, to then, get some energy and to give off some gas and fluff up that bread. Anyway, so what we're going to do on this side is we're going to be looking at aerobic cellular respiration, that whole process. We're going to draw some dotted lines. So if you would draw a dotted line that goes along with this one, that goes down to pyruvic acid, and then across like this, and then back up to the line over here, that would be fantastic. And then the next one I want you to do is there is this line right here, which I want you to do a dotted line that goes right up to where 2 acetyl-CoA are, and then go underneath where it says NADH2, which is also, of course, also known as NADH plus H pluses. Okay, and connect that up over here because we're kind of doing these chunks. Remember, we talked multiple times about there being 4 chunks that occur during the aerobic cellular respiration process. So we've got glycolysis right here, you've got your link reaction going on here, you've got your Krebs cycle, also known as the citric acid cycle, because here's a shock, there's citric acid in it. And then, last but not least, would you do a dotted line that goes across right here, okay? Because down here, we've got that oxidative phosphorylation that has the 2 parts to it that we're going to put together on here. All right, so let's start back up here. And let's just go right in a row. We're going to start with glycolysis and do reactants and products, also known as inputs and outputs. Okay, and so we're just going to fill in these boxes as we go. On this side, what we're going to do is we are going to be putting in what it is. So this is our glycolysis. Okay, which, by the way, there's more on page 166 and 167 if you'd like some more info. More detail than you need, but it's still there for you. This one, glycolysis, as you know, because it's done by both aerobic cellular respiration and anaerobic cellular respiration, it requires no oxygen, okay? It also occurs in that gooey stuff of the cell, that cytosol, okay? So when we look through this process, you've got glucose that as it's being broken apart, because it's a C6 molecule or a 6-carbon molecule, into 2 pyruvic acids, so it's getting broken in half, right, and converted. It's going to need some ATP to get that done, but it's also going to be able to use some ADP and some PI to make some ATP, okay? That's why when we talk about glucose being broken down into pyruvic acid and glycolysis getting you 2 ATPs, it's a net of 2 ATPs. So you're going to take 2 ATPs to get the process started, okay? But in the end, you're also going to produce 4 ATPs. That leaves you a net of 2 ATPs throughout that whole entire process. Not only that, you're going to get, you know what? I want you to grab another color because I'm a pain in your tuchus. So, remember your NADH? Okay, I think back in the day we had, another color for that. I'm going to use a little teal here. So NADH, also known as NADH plus H pluses, okay? That's the other thing that's made during this process of glycolysis. So let's look at our inputs and outputs. If you look over here, you've got glucose coming in, so a C6H12O6. You also have, notice, 2 ATPs that need to be utilized to be able to run the process, okay? You're also going to need, so since you're going to make some ATP here, you're going to need 4 ADPs, adenosine diphosphate, plus 4 phosphates, or OPO3s or PO4s, okay? The other thing is, if you're going to make some NADH plus H's or NADH2s, you're going to need 2 NADs, okay? So, these are our inputs. These are the things that have to come into this process to get it to run. What comes out over here is that you're going to get some water out of this. It's not pictured on here, but you're going to get 2 water molecules coming out of this process. You're going to get 2 of those NADH plus H pluses out of that process. Okay. You're going to get 2 ADPs and 2, I'm getting running low on space, so I'm going to say and, 2 PIs, right? And then you're going to get 4 ATPs, okay? And then last but not least, the big one, the big goal on this one, okay, is that you're going to get 2 pyruvate. And remember, pyruvate is the same thing as pyruvic acid. It's just 2 words for the exact same thing, okay? So, glycolysis, no oxygen required. Every organism does it. Doesn't matter if you're a prokaryote or eukaryote. You do it in the cytosol. Takes glucose, breaks it down into 2 pyruvic acids or pyruvates, and that gets you 2 ATPs to be able to utilize for energy, and then 2 of these lovely, remember, electron taxis of the NADH plus H pluses. So, once we've got this pyruvate or pyruvic acid, okay, it's going to be converted during the next part, okay, the link reaction. Okay, that's going to be followed by the Krebs okay, or citric acid cycle. And then that's followed, of course, by, and citric acid cycle, by the way, is on page 168. The link reaction is also on page 168. It doesn't call it the link reaction, but the information is there. And then the last part, of course, is oxidative phosphorylation, which is on page 172. Now, to do all of this for aerobic cellular respiration, you not only need some oxygen in here at times, but you also need to have, of course, the lovely organelle known as a mitochondrion. So in the mitochondrion, overall, and then we'll talk about what parts of the mitochondrion for each one of these, okay? So let's start with that link reaction. The link reaction is one part that does require oxygen. It happens in that inner membrane space. So remember that itty-bitty living space, okay? In that itty-bitty living space, what you can have happen in this little tiny space is you get a conversion of this pyruvate, also known as pyruvic acid, into acetyl-CoA, okay? So while doing that, you're going to take some and have some inputs and outputs, one of which, of course, is the 2-pyruvate. You're also going to need 2 coenzyme A's. These 2 coenzyme A's are not only an input, but they're also going to be an output down here because they cycle back up and around because it's an enzyme. It gets reused, 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 right? And so we've got 2-pyruvate, 2 coenzyme A's, and 2 NADs, because we're going to get some more of those NADH plus H pluses. So those little electron taxis are going to come up again right here, okay? So what you're going to get out of that is you get 2 of your acetyl coenzyme A's, okay? That's what you need to go into this Krebs cycle. You're also going to get a couple of electron taxis, NADH plus H pluses. Okay? So you're going to get some electrons that are going to be able to go down here. Notice this big arrow. You've got the NADH plus H from here, the NADH plus H here, the NADH plus H here, and the FADH2s from here that are going to go way down here into that oxidative phosphorylation. They're going to drop off the little electrons because these are all taxis, okay? And there's their little highway. I know, cheesy. All right, so we've got 2 NADHs. We're also going to get, notice, we got to be able to get rid of, we're going from a 2-carbon molecule from a 3-carbon molecule. So if we're going to go from here to here, we've got to go and get rid of some carbons. You don't cough up carbons, so instead you cough out carbon dioxide, so 2 CO2 molecules, okay? The next part, of course, is once you've got this acetyl-CoA, it is going to go across that membrane and into the next place where the Krebs cycle occurs, which is that gooey part, that matrix. The thing about this is there's actually no oxygen required to do this process. It's just sandwiched on either side. For aerobic respiration to occur, you just need the link reaction and you need oxygen for oxidative phosphorylation. Okay? So now, we've got the Krebs cycle. And so you start with some acetyl-CoA. It then gets converted. We got some citric acid. We're going to need to start. Notice we're building a 6-carbon molecule, and we're going to break it down. And so we're going to get a 5-carbon molecule. So we're going to need some CO2s coming out to get rid of those. As we do that, we're going to be able to pick up some electrons and put them on some taxis. And we're going to be able to, as we go from a 5-carbon to a 4-carbon molecule, and we got to get rid of some more carbons. Okay, when you break those bonds, you're going to be able to have energy to make some ATPs. Okay, so what that means is you're going to have an input of 2 acetyl coenzyme A's What that means, though, is these coenzyme A's are to come in here and get it to do its job, to make it become part of this process. So these two coenzyme A's are going to leave this process, be an output, and go back up here and work on this again. Okay, so there's like a little mini cycle right here for coenzyme A, okay, that comes around and around, back and forth. The next thing is you're going to be needing some NADs, because those NADs, and you're going to need 6 of them, because you're going to get 6 NADH plus H pluses, those little electron taxis, being formed and being an output. So here they are right here, okay? You're also going to have, oh, see how I am. As you can tell, need another color. I'm going to use orange now. Sorry, there we go. And so you're also going to make some other electron carriers or electron taxis. The two FADs are going to come along so that you can then pick up electrons and hydrogen ions to be able to form FADH2s. Okay? And so if I remember right, we're using this orange like what we did way back when we did the big version of this back a few pages in your IMB. Okay? It also is going to be one of those electron taxis, going to take the little highway down here to oxidative phosphorylation. Okay? The next thing is we're going to need some waters to do this, 6 H2Os. Okay? Also going to need to be able to make some ATPs. So if we're going to make some ATPs, you got to have some ADPs, and you need to have some phosphates. Okay? So we're going to get out of this process, if you notice we've got some here, and some over here, we're going to get 6 CO2s coming off, okay? And we're going to get 2 ATPs being formed during this process, okay? So here they are right over here. So the big goal of this is the Krebs cycle, which occurs in the matrix that doesn't require any oxygen, just cycles around taking carbons off of the molecules, breaking bonds, releasing electrons and sucking up some hydrogen ions to be able to transport some electrons, as well as making some energy right here. But the big thing, we get lots and lots of energy from doing aerobic cellular respiration. Remember, up here, these guys are only getting 2 ATPs, okay? No matter which kind of anaerobic, we, on the other hand, are going to get 38 ATPs. Problem is, look, we only have 2 net right here, and we've got 2 right here. That's 4. We need to get to 38. In fact, it gets a range of 36 to 38. So that means we need to make more ATP, and that's where this last process comes in. It's the part where you're making all that extra ATP, that big bang we get for that buck. Because if you think about it, anaerobic cellular respiration is like having $1. Having 1 glucose molecule, which is $1, and you get 2 cookies for that, okay? Aerobic cellular respiration uses the same freaking dollar, 1 glucose right here, and what we're going to end up in the end is we're going to end up with 38 ATPs instead for the exact same dollar. So we more efficiently use the exact same molecule, but you got to add in a lot of processes to get that done. So we end up adding link reaction so that we can do the Krebs cycle, and then we use all of these little electron taxis from all these up here. They're going to all come down here. So now when you add all these up, you're going to get 10 NADH plus H pluses, okay, coming in as that little electron taxi to drop off hydrogen ions and drop off electrons, right? So you're going to get both of these little hydrogen ions as well as these little electrons, okay? You're also going to get 2, okay, FADH2s, which are the other electron taxi, to drop off more electrons and more hydrogen ions, okay? What that means then is it's a nice cycling process so we can run these again. Notice these arrows going back right here. We're going to get these NADs and FADs that are going to, the 10 NADs and the 2 FADs are going to go back up here and be reutilized, okay? So what's been dropped off to this process, to this oxidative phosphorylation, where O2 is the final electron acceptor molecule, when that happens, sorry, I was just checking the time. It is now 12:15. So if I don't get this done in 15 minutes, I got to pause and I got to go run and turn off the alarm and stuff. So there you go. It happens, of course, using two things. Remember we talked about this when we did our little T-chart. We talked about how electron transport chains need to use membranes and they need to use an itty-bitty living space. So in this case, we're going to be using the cristae, which is that membrane. Okay, that's where the electron transport chain portion of oxidative phosphorylation is going to occur. And then you're also going to need the matrix. In that, you're going to need a source of hydrogen ions that can be pumped across. And you're going to need that intermembrane space, that itty-bitty living space, okay, to be able to build up that electrochemical gradient, also known as a membrane potential. In other words, that's the chemiosmosis part. Osmosis. Okay, so in this visual, what you're getting is you're getting all of these little electrons, okay, that are being dropped off here. This right here is the electron transport chain. Both NADH plus H and FADH2, remember, are dropping off these electrons. They're also dropping off some hydrogen ions, okay? When that happens, when they do this, remember, that's going to produce an electrochemical gradient. It does this by pumping. Remember, those proteins are embedded in the membrane are going to pump from the matrix by pumping hydrogen ions from the matrix into that itty-bitty living space, that inner membrane space. Okay? Because of that, you're going to get a charge. And because of that, there is this process of doing chemiosmosis using ATP synthase. Okay, it's kind of like if we made a little bulb here. This right here is going to be your ATP synthase. Okay, so this chemiosmosis uses ATP synthase, and these little hydrogen ions are going to rush back through here. When they go in out here and to the matrix, 32, okay, 32 to 34 ADPs and 32 to 34 phosphates are put together to make 32 to 34 ATPs. Okay, so you're going to get 32 to 34 ATPs out of that process. Okay? The problem is, at the end of this electron transport chain, out pop all these stinking electrons again. Darn it. So these are highly reactive. Lovely oxygen that we get, we combine those together with some of these lovely extra hydrogen ions that are out here in the matrix, okay, are going to get you some lovely water that's nice and safe for us to handle. So we don't have dangerous electrons, we don't have oxygen ripping things off, and we don't have some extra hydrogen ions doing some funky things either. We make some water out of that process. So we get ATP and we get some water. So let's look at the inputs and outputs, okay, to this process. We've got these guys, these electron taxis are dropping off electrons to the electron transport chain. We're also going to need some more hydrogen ions. We're going to need some oxygen to be able to be that final electron acceptor molecule. We're going to need 32 to 34 ADPs. We're also going to need, of course, 32 to 34 pi's to go with that, so that we're going to end up with, thank you very much, if you look at this, plus those lovely little electrons, okay, are going to get you those 6 waters, okay? You're also going to get 32 to 30 p from that process. What that means with all is said and done, after you use this, remember right here, you're using the ATP synthase to be able to do this, okay? Once you're all said and done, you're going to end up with this process right here. We can write that more simply by saying we start out with glucose C6H12O6 plus 6 O2s will get you 6 CO2s. Remember, these are the oxygens that are needed for the link reaction, as well as oxidative phosphorylation when it comes to being the final electron acceptor molecule. We're also going to get some 6H2O. All I got to do is breathe out to feel that. And then we're going to get approximately 38 ATPs. Usually about 36 to 38, depending on the efficiency of the process. Life is not always perfect, so we're not always going to get 38. What that means is you're going to get an equivalent of, so this much ATP will get you an equivalent of work in the cell that's equal to 686 kilocalories, okay, so 6,860 calories per mole of glucose. Okay, way more bang for your buck for the exact same one glucose molecule that those anaerobic ones got only 2 for. But it takes a lot of work to get there. Okay, it's getting close to the alarm time, so we'll see how this goes. I'm going to turn it over onto the back so we can do photosynthesis now. This is where you're going to need some red for ATP. I think what I'm going to do is I'm also going to do another color since I'm a pain in your tuchus. So I'm going to want you to do another color for I think I'm just going to go tan on that one. Ooh, we could do gold. No, how about hot pink? No, that's too boring. Dark blue. Okay, so we're going to be needing the light blue because I'm going to use that for the linear electron flow that goes through this first process. I'm going to have pink so I can intersperse over the top of that or put on over the top of that the cyclical flow that can happen when this plants need more ATP, so we're going to talk about that. But we're also going to need the ATP color that you have and your NADPH color, okay? So I think what I'm going to do is I'm going to use dark blue. I don't think that's the color I used back a few pages and stuff. No, I take that back. Okay, I'm indecisive. It's after midnight. So I'm going to use purple because I want to. All right, so let's look at this. Photosynthesis. Photosynthesis, of course, starts with light energy. Light energy right here, my little smiley face sunshine, because it's such a fun process right there. That light, of course, is hitting onto the main photosynthetic organ of the plant, known as, shock, a leaf. Now, the only time that there's an exception to this, this is a broadleaf, right? So like a deciduous plant or a broadleaf plant. Both those two are going to be this way. The exception to that would be if you're talking a cactus, it's actually their stem that does photosynthesis because all those little spines that stick you, right? All of those are actually the modified leaves. So it's either going to be the leaf or the stem, but it's going to be the same parts within that plant. So if we look at a typical cross-section of a leaf, it doesn't matter if it's broadleaf like this or a needle for, say, a deciduous, or excuse me, a conifer like a redwood tree, okay? You're still going to need to be looking at the mesophyll. Remember, meso is middle and fill like filling, okay? That mesophyll, okay, is where the light-dependent and light-independent reactions occur. So the first part is we've got the mesophyll being this, not that part right there, okay, but being this part right here, this middle section is that mesophyll. Remember, there's also, let's just make some other things that are in here. We have these guard cells right here. Remember, the guard cells are the ones that inflate and deflate with water to be able to open and close this hole right here, which is the stoma or stomata, okay? And then you, of course, have this surface layer, which is the epithelium, the epithelial layer. And then on the surface of that, on the top, you have that waxy cuticle. Okay, over the top of that, on both the top and that bottom down here. Okay, so the two parts that we're interested in the spongy mesophyll are these ones. Remember, these are those columns. Okay, so those are the palisade mesophyll. Okay, and then you also have right here the spongy mesophyll. Okay, because it's like a sponge. It sucks up all the gases that you need, like carbon dioxide as doing this process. It also helps get rid of oxygen. The only downside to that, but actually a benefit too, is water gets lost out of there. So the thing is, water being lost out of there can help do transpiration, so it'll actually help water move up a vascular plant through their xylem, because xylem is dead tissue. But if it happens too much, the plant starts to dry out and can die. So that's why you have, remember back in those other notes, when we talked about the C4 plants and CAM plants, which compensate for needing to close those stomas using those guard cells, okay? So we're not going to review that here. That's just in those photosynthesis details notes. This one right here, we're going to be talking about the photosynthesis process that is the C3 version of that process, okay? Palisade mesophyll, you might also hear other names for this. I've seen other ones on different tests, okay? They're also known as just the palisade, okay? And they're also known as, aka, what's known as the parenchymal cells of the palisade mesophyll. Okay, so this is just a little more specific. Sorry, I'll make sure that's on the screen. Okay, and so this is just another way you might hear it as parenchymal cells sometimes. Okay, so when we talk about that, of course, most of them is where this is going to happen. So here's your palisade mesophyll. It is a columnar cell. Okay, most of it happens there is some photosynthesis that will occur in these spongy mesophyll cells. There is some, but the majority of it is happening in this lovely palisade mesophyll, this columnar cell. Within that, there are these little guys right here, okay, which, I'm just going to go crazy on you here. You never know when I'm going to pull out another color, right? You've got these lovely green things roaming around in that cell. Those green things, of course, are right here, that chloroplast. And remember the chloroplast, looking at the time, I'm going to have to pause here in just a second. Right after we do this, I'm going to take a pause and then we'll come back and do this process because I got to go check the alarm. You have, of course, these little things right here. Inside these little sacs is that itty bitty living space again, the thylakoid space, okay, that is inside a thylakoid sac, okay, which is made up of thylakoid membrane, okay, and that's thylakoid membrane, whether it be these connected lines or whether it be these little sacs right here, okay. So all of this, these little lines right here and these little sacs are thylakoid membrane with embedded embedded proteins. So those integral proteins, it has peripheral proteins. Remember when we talked about transport proteins in the phospholipid bilayer? Those kind of things. There is also, of course, this space out here and in here, any open space in here that surrounds these thylakoid membranes is going to be the stroma. Stroma. So that lovely stroma with the R, that's going to be that goo that's inside that chloroplast. So remember, it's only these little sacs and membranes that actually have a pigment. I'm coloring like pukaka, but you understand what I'm doing here, right? That's where it's embedded with the proteins that include pigments. Okay, gotta run, gotta go check. And I will be right back because I gotta, oh my goodness, I hope I don't set off the alarm.